It is a real honor to be here. It's really going to be a tough act to follow the 500,000 genomes, and always a tough act to follow Robert. But I am going to talk to you about the proteomics data that's been recently generated and released in the UK Biobank. And Rory was kind enough to give an introduction to this today, so I think I can go over it very quickly. There are 13 pharma companies that were involved in generating this data. And Rory also called this out, but this is one of those things. We kicked this off in 2020, but this is an idea that we had been talking about for quite a few years. And really, this is something that was really dreamt up by and really pushed and championed by Chris Whelan, who was at Biogen, now at Janssen. And if it weren't for Chris's leadership, this is something that we couldn't have done. So the consortium went through a lengthy process, evaluated multiple proteomics platforms, and we eventually settled on the Olink platform. So we measured 3,000 proteins on the Olink Explore platform in about 57,000 samples in the UK Biobank. So for people who are not familiar with the Olink platform, this is a proximity extension assay with an NGS readout. And that technology enables us to do multiplexing of proteins all on the same plate, so very large-scale proteomics measurements at a very small volume a very small sample volume with great accuracy. So we received the last batch of the proteomics data in mid-2022, and I'm pleased to announce that we had 3 flagship publications accepted in Nature. The publication on the left there is the consortium paper. We looked primarily at common disease associations, as well as starting to scratch the surface of associations with health and disease, and how those are influenced by the plasma proteome. Something which I have been calling the rare variant paper, but now need to start calling the rare coding variant paper. This is from colleagues from AstraZeneca, Biogen, and Janssen, looking at the exome sequencing and how that influences levels of proteomics. And then finally, a paper by our colleagues at Decode and Amgen doing a large-scale comparison of different proteomics platforms. So I'm a genetic epidemiologist by training, so both from a genetics perspective and an epidemiology perspective, I'm really interested in how this kind of data can help what we know about genetics and how we use genetics, but also how this type of data can be used to teach us about human biology and human disease. So there are a number of different ways I think that the proteomic data has been and will continue to be useful in this cohort, and these are just some of the examples here. And I certainly won't have time to talk about all of these today, but I do want to talk about a couple of vignettes from the publications, as well as a couple of vignettes from other publications that have come out of other groups. So I'm first going to focus on some of the genetics findings that have been found both in our flagship papers, as well as some of the work that's starting to come out with the whole genome sequencing. So turning back to our flagship consortium paper, and again, this is looking at common genetic variances and how those influence the plasma proteome. What you can see there on the left-hand side is the map of all of the pQTLs that we identified. That is, for people who are not familiar with this, that is the map of the genome, so chromosomes 1 through 22. Each one of those dots is a pQTL. So we were able to identify 14,000 pQTLs in about 2,400 of the proteins. About 2,000 of the proteins contained a cis pQTL, and 27% of those were novel. Cis pQTLs are those where the association is located within the cognate region of the gene. What's really, truly transformative about this data is that we were able to identify 10,000 trans pQTLs, and almost 90% of those were novel. So this just shows the power of discovery with a sample size this large. We were able to identify novel trans pQTL associations. We also did look at the pQTL mapping for the non-European ancestries, found that it was largely consistent, but were able to identify some potentially interesting signals we wouldn't have otherwise found in Europeans. So the first example I do want to talk about is a non-European pQTL mapping. And again, while these were largely consistent, there are a few examples. So this is a region called ANXA4. What I'm showing on the bottom half of the plot there is the association in this region in Europeans. And of course, there is a quite robust signal there. It's only not considered significant because we did increase the threshold for multiple comparisons. You look in the African ancestry population, which is about 3% of the sample size, is the European. And you can see we have a much more robust, strong association in this region. And there's a couple of more examples of this in the publication. And what we have largely discovered is that we find these novel pQTLs, or much more robustly associated pQTLs in the non-European population, often due to differences in allele frequency. And I think this really highlights something that has gotten a lot of press in the genetics industry and something I'm very passionate about, which is really both the value and the need to be generating this type of data, both genetics and proteomics, in non-European populations. So if we switch now to the rare coding variant paper, so this is work from Ryan DeCinza as well as other colleagues from AZ, Biogen, and Janssen. Again, I'm going to show you the same pQTL map, except for this is now for very rare variants with an allele frequency of less than 0.1%. They identified about 5,000 different variants, and interestingly enough, if you look at the plot here, almost 80% of those are novels, so we would not have picked them up using GWAS alone. So this just shows you the value of collecting exome sequencing as well. I think the other thing that's really interesting is if you look at the direction of effect for these associations for the coding genome, the majority of the associations that you find decrease levels of protein, so you're looking at things that are more likely to be loss of function as opposed to things that would increase the protein. And again, very similar to the example that Robert just showed, I think one of the use cases that we found particularly interesting with this work is being able to incorporate the proteomics data into your collapsing test. So here, you could take missense variants that are quite rare, that have an effect size, that reduce protein by a certain amount, and you can incorporate those into a burden test. And you're able to see that at least for certain traits and certain phenotypes, you do see an increase in power. So this gives you a little bit more power as we are doing our genetic identification and discovery. And of course, I would be remiss if I weren't going to talk a little bit about the whole genome sequencing. So this is work that came out in bioRxives from the Exeter group about a month ago, looking at what was available at the time, which was the first 1,500 proteins, and it's in about 20,000 people who had whole genome sequencing. You can see this is all of the single variant cis pQTL tests across the genome for the whole genome sequencing. And we're talking about p-values that are so strong here that you actually can barely see the significance line, which is pretty cool in and of itself. If we are to look at the effect sizes between the coding and the non-coding genome, you actually see the distribution of effect sizes is pretty comparable across the two groups. But something that the authors noted in this paper that I also thought that was really interesting was how the effect sizes are distributed. So if you remember on the last slide, I mentioned that in the coding genome, the majority of the associations that you find decrease the levels of protein. If you look in the non-coding genome here on the left-hand side, you can see that the distribution is actually pretty even. So these variants are just as likely to increase protein levels as decrease protein levels. So I think that's a pretty fascinating learning in and of itself that you see these differences between the two genomes. So at this point in time, I've talked largely about genetic results that we've used this data for, but of course the value of the UK Biobank is the deep and rich phenotype that we data that we have as well, and there's amazing things that you can do with this data even outside of the genetics. And of course, we were already lucky enough to hear today the early career researcher of the year discussion from Julia and Claudia Langenberg's group, which is fantastic because it always takes me a really long time to explain this slide, and Julia did a much better job than I would have done. So again, this has been a really invaluable resource to be able to start thinking about what kind of proteomics can predict disease. And certainly we have diseases out there for which we know it is hard to identify who's going to go on and get this disease. So incredibly valuable to have that data. And the other thing that we know is that this data does a better job of predicting some diseases than, say, something like the circulating biomarkers does. The last example I'm going to show is from Aaron Smith and our colleagues at Takeda, and this is looking at levels of neurofilament light chain and whether those are associated with sporadic motor neuron disease or ALS prior to diagnosis. So levels of neurofilament light chain are known to be associated with genetically determined ALS, but there's less known about sporadic ALS. And so what Erin and her colleagues were able to do were to identify the sporadic cases of ALS in the UK Biobank, and then they were able to use the EHR data and determine whether or not neurofilament light chain levels were higher the closer you were to diagnosis. And you can see actually that there is quite a steep increase once you get close to diagnosis. So it's a pretty profound looking effect. And in fact, you can then use this data to stratify and be able to stratify, and you can see very clearly that the people that are in the highest decile of neurofilament light chain have a very, very high risk of going on to develop ALS. And this, again, is the first example, and I think this really highlights the overall value of UK Biobank, that we have the genetic data allowing us to figure out if an ALS case was sporadic or genetic, that we have the electronic health record data that's longitudinal to be able to do work like this. And so hopefully I have been able to convince everybody today the value of this data. We are so excited that it's available and out there in the world. There have already been so many fantastic papers, and we're really looking forward to seeing what additional work comes out of this data. And so with that, I'd like to thank everybody in the consortium. I'd like to thank the UK Biobank for being such wonderful partners putting this all together, colleagues at Pfizer, and of course also colleagues at Olink for working with us to generate this data set. Thank you.